Hello, my dear lab students. This is Dr. Kat Vlies, and we're going to start the nervous system with first an introduction to the hierarchy of the nervous system, and then we'll dive into the different cells that make up the nervous tissue. So we tend to refer to all of this. This is the study of neurology. So by now you've probably covered the hierarchy of the nervous system in lecture, so I'm going to go over this pretty quickly. Remember that the central nervous system is only made up of the brain and the spinal cord, so everything else is part of the peripheral nervous system, which in its turn has two major divisions, the sensory division and the motor division, which are made up of sensory neurons. You may know them better as afferent neurons, and the motor division, or the efferent neurons. Within the motor division, you then have two different kinds of motor neurons. The somatic motor neurons belong to the somatic nervous system, and their effectors are only skeletal muscles. Be sure that you remember this, that somatic motor neurons only innervate skeletal muscle, while all of the other motor neurons belong to the autonomic motor nervous system. Actually, it would be better for us to add in here motor and here autonomic motor nervous system. This will remind you that we're dealing with the motor division. But the autonomic nervous system you can call the visceral motor nervous system, and some of you might like that better because that'll remind you that it innervates our viscera, right? Which are, remember, our digestive structures, our heart, our lungs, our blood vessels, basically anything that's made up of smooth muscle. Of course, our heart is made up of cardiac muscle tissue, and then all glands. And by glands, we mean not just glands that make hormones, but that secrete a variety of things, from tears to saliva to mucus, and the list goes on. And then in lecture, you'll learn a little bit more about the subdivisions of the autonomic nervous system. In AP2, you'll learn a little bit more about the so-called enteric nervous system and the digestive system. We're not going to spend any significant time discussing the enteric nervous system in AP1. So let's dive into the nervous tissue. The nervous tissue is made up of two cell types, the neurons, and remember, we can also refer to the neurons as nerve cells. Don't forget that. Realize that nerve cells are not at all the same things as a nerve. A nerve is actually an organ. We'll look at it later on. A nerve is an organ made up of many bundles of axons. There we go. And so the neurons are one type of cells that we find in the nervous tissue. And then we have all these so-called supporting cells, or glial cells, or you can even call them neuroglial cells. And as their nickname says, they literally support the neurons. Now we must make a separation between the neuroglial cells of the PNS and the CNS. In the PNS, it's primarily the Schwann cells that we'll focus on, or that we need to pay attention to. They make the myelin sheath, that insulation layer around the axons. In lecture, you may learn a little bit more about these satellite cells. We don't really know much about them, so we're not going to really address them in lab. So in the central nervous system, notice the four different supporting cells that we see there. And of those four, it is the oligodendrocytes that make the myelin sheath. So a very different cell, right? So in the PNS, it is the Schwann cells that make the myelin sheath. In the CNS, it's the oligodendrocytes. Make sure you keep that separate. Another cell that we're going to bring up, especially when we get to the brain, is the ependymal cells, which line the ventricles and the central canal of the spinal cord. Now, what are these structures? These are the central canal and the ventricles. They're filled with cerebrospinal fluid. So your ependymal cells play an important role in the helping of the making of cerebrospinal fluid, but also in keeping it moving. So here we're taking a closer look at the Schwann cells. Remember, they're only present in the peripheral nervous system. So here we're looking at an axon of either a motor neuron or a sensory neuron. And notice how a single Schwann cell continues to wrap around the axon. It's as if you're wrapping an air mattress around a broomstick and you're pushing out the air of the air mattress. And ultimately, that you can't always get that very last little poof of air out, so that's where all the organelles and the nucleus of the Schwann cell accumulate. Now, we don't just have one Schwann cell. This is a little bit misleading, perhaps, this picture. Of course, you would have a second one right here around the axon and another one here around the axon if we continue to draw the axon further like so, right? So here is our axon. So this is a Schwann cell, this is a Schwann cell, and so on. And in between these Schwann cells, we have these gaps that we call nodes of Ranvier. This is where we have large amounts of voltage-gated ion channels. And that is, of course, where we do not have any insulation. More on that in lecture, but this is what the Schwann cells look like. And they are your ordinary cells. They can divide, just like many of the other cells in our body, unlike neurons, which cannot divide. And here then we are in the CNS, where we have 4 different kinds of supporting cells. I want to point out how the myelin sheath is formed in the CNS. So here we have, for instance, an axon. And it's this cell with all these long arms that helps form part of the myelin sheath. So we're going to need multiple oligodendrocytes to wrap the ends of their long arms around different axons. So a very different kind of cell that forms the myelin sheath in the CNS. And then, as I mentioned before, we're in the brain also going to talk a bit about the ependymal cells. Here you see them lining a ventricle. A ventricle, or it could be the spinal canal, the central canal of the spinal cord. This is where the cerebrospinal fluid is located. And they, depending on where they are, it might vary some. You'll learn more about that in lecture, but they have cilia in most places that help that cerebrospinal fluid moving. It's very, very important for our cerebrospinal fluid to move. As we'll learn in our discussion on the brain, we constantly make cerebrospinal fluid and we constantly remove it. So it's a continuous process of making and draining the cerebrospinal fluid, and it needs to keep moving. So remember, nervous tissue is made up of the neuroglial cells, which we can separate between the PNS and the CNS. We just did that. And then the neurons. So let's spend a little bit of time looking at the anatomy of a neuron. A neuron is made up of a cell body, and it's going to have some dendrites, sometimes, or in this case, they're coming off the cell body. That can vary depending on the shape of the neuron, as we'll see in a little bit. And we have a major branch that you see here, which is called an axon. Now, since we have so many branches coming off the cell body, so many poles coming off the cell body, the structure of this particular neuron is described as multipolar, meaning it has many poles coming off its cell body. All right. Okay, so dendrites, the beginning of the axon is called the axon hillock, and then the axon will eventually make it to its destination, which could be another neuron, as illustrated here, or neurons. And then the end has axonal terminals that synapse with the next neuron. Or we can look over here. These are the axonal terminals of a presynaptic neuron. We have more here, we have more here, right? So these are neurons that sit prior to our synapses, for instance here, or here we're focusing on this synapses illustrated here. In lecture, you've learned how we have synaptic vesicles filled with neurotransmitters that are released into the synaptic cleft, and then the neurotransmitters bind to their receptors on the postsynaptic membrane. Now, it is possible for instance, a somatic motor neuron or an autonomic motor neuron to synapse with an effector, right? So we wouldn't have neurons here. Instead, we would have muscle tissue or some kind of a gland. It all depends on what kind of a neuron we're looking at. The fact that we are synapsing, the fact that this neuron is synapsing with other neurons, and even here other neurons, that is already kind of telling us, this is probably, since it's multipolar, either an interneuron or a motor neuron. Now, the fact that we see Schwann cells tells us that this has to be a motor neuron. Now, autonomic motor neurons consists of two in a row. So it's, if the fact that we see another neuron here where synapses is occurring implies that this is going to be some kind of an autonomic motor neuron. I'll explain better why we would see two motor neurons in a row in the autonomic nervous system. Okay, now in the cell body, we find your traditional organelles, with the exception of centrioles, which is why neurons cannot go through a mitotic division. But what's really important for you to also be aware of is that the rough endoplasmic reticulum, which you see here in the purple, and the Golgi bodies are very well developed. And just so that you see this better, I'm going to put little dots everywhere along here, this rough endoplasmic reticulum, to remind you that it's studded with ribosomes, and of course that means protein synthesis, while the Golgi body can package up the proteins. We refer to the rough endoplasmic reticulum of neurons as Nissl bodies. So be sure that you recognize this term, Nissl bodies. That is used more often than the rough endoplasmic reticulum. We also see here mitochondria. There's going to be lysosomes and your typical organelles present, smooth endoplasmic reticulum. But the reason why I'm really emphasizing the Nissl bodies and the Golgi bodies is because they are totally absent from axons. And so therefore axons cannot go through protein synthesis. So here again, we have a nice image of a multipolar neuron synapsing with yet another multipolar neuron. This time our axon is not myelinated, which is perfectly normal. The little branches here are referred to as telodendria. Don't worry about that term. We are typically just pointing out these axonal terminals. Here they're calling them the synaptic terminals. There's all kinds of slightly different names for them. Okay, so this brings us to how we classify neurons. And this is kind of important, as you've noticed in your lecture as well. You can always classify things based on what they do. That's one way of grouping things, or we can group them based on what they look like. So we're going to do that with our nerve cells as well. So if we look at what neurons can do, they can either sense the environment, they can relay that sensation to the neurons that then tell the effectors what to do. Remember, effectors include all muscles, meaning skeletal, cardiac, smooth muscle tissues, as well as glands. And as I've mentioned before, there are two types of motor neurons, your somatic and your autonomic motor neurons, and they have different effectors. Somatic motor neurons innervate skeletal muscles only. So these are, when you're asked what is this functional neuron, and it's pointing to some picture, you need to pick from these three choices. If, on the other hand, you're asked to identify a neuron based on its structure, so what kind of a structural neuron is this, you have these three choices. And more than likely, it's going to be one of these two. Bipolar neurons are quite rare. You already know what a multipolar neuron looks like. And we'll see what a unipolar neuron looks like on the next picture. A bipolar neuron is indeed a neuron with just two poles. So it would be as follows. It would have a cell body with a process here and a process there, like so. So this is one major dendrite that then splits up into smaller dendrites. So your action potential would travel in that direction. So you have just two poles, or it's bipolar. So now we can take a closer look at how action potentials travel in our body with the help of all of our different kinds of neurons. So what we're seeing here is that we're looking at sensory receptors. Perhaps this is representing the skin. Let's indicate that, right? This could perhaps be the skin with, the root hair plexus, as we've learned about, which are really just the dendrites of our sensory neuron. If there are some layers of connective tissue cells wrapped around, it could be a Pacinian corpuscle, right? Etc. Of course, we don't have just touch receptors in the body, but if we're looking at the skin, this could be a good visual for us. So this is where our sensation occurs, and that the mechanical stimulus of a bug crawling on you or somebody touching you is then converted into an action potential, right? And it's going to fly up this neuron. And notice the funny shape of this neuron. Right here we have a cell body, and notice that the cell body has just one short little pole right here that immediately splits. But it just has one pole or one branch. So this is a unipolar neuron. This sensory neuron, that's the functional classification. So this is a sensory neuron. You may call it an afferent neuron. And what is its structure classification? It is unipolar. These are not synonymous terms, by the way, right? We're looking at what this neuron does. It carries the sensation into the CNS. All of this right here represents our CNS. Let me use a better color. Let's use red. So all of this is CNS. All right. Everything outside is PNS. So we're literally carrying our action potentials into the CNS with the help of our sensory neurons. And they happen to have the shape of a unipolar neuron. Yes, right, a structural neuron that is unipolar. The cell body right here is always going to be left. Cell body is left outside the CNS. And so if we have lots of sensory neurons going into the CNS, they're all going to leave their cell body sitting outside of the CNS. So they'll start accumulating here, and we'll get this big blob of cell bodies, and that's what we call a ganglion. And we'll see that term more often as we move on. A ganglion is a collection of cell bodies outside of the CNS. Now notice that the axonal terminals of your sensory neuron are inside of the CNS. And more than likely there, they're going to synapse with an interneuron, which happens to be multipolar. You see it? There might be many interneurons, or we may not even have an interneuron. In our very fast reflexes, these axonal terminals, or some of these axonal terminals of this sensory neuron, will synapse immediately with a motor neuron, right? So then our action potential flies immediately from our sensory neuron straight into our motor neuron to its effector. Now let's take a look at the effector here. The effector here is skeletal muscle tissue. I'm sure you recognize this, the striations, the long cylindrical shapes of the cells. And so therefore, you know, this has to be a somatic motor neuron, right? Only somatic motor neurons innervate skeletal muscle tissue. And notice where its cell body is located. It is located inside of the CNS. Now, let's say we were looking at innervation of cardiac muscle tissue or smooth muscle tissue, right? And glands. Let's say we were looking at the other effectors. Well, then we would have, again, a cell body of our motor neuron in the CNS, but this neuron wouldn't be quite that long. It wouldn't make it all the way to its effector. There would need to be a second one, or many second ones. I'm just going to draw one. And so we have literally two motor neurons in a row. So this is going to be, these are representing autonomic motor neurons. So you have two in a row. And notice, therefore, that once again, we might end up with multiple cell bodies in the PNS, right? Just like we had here in the case of our sensory neuron. This time, this ganglion would be an autonomic ganglion. But in general, most cell bodies of motor neurons, as you can see, are going to start in the CNS, right? So your action potential goes from your receptor up your sensory neuron through the central nervous system via interneurons and then out to its effector via either somatic motor neurons or autonomic motor neurons. This is a really important diagram for you to get. Notice too that your sensory neurons are unipolar, your interneurons and your motor neurons are typically multipolar as well. Are there bipolar neurons, and are there really bizarre looking interneurons in the brain and spinal cord, especially in the brain? You bet. But like I said, bipolar neurons, we'll see some of those when we study the eye. So in the next few slides, I'm going over what I have been saying already. Here you're looking at the sensory neurons, which are these guys, and they're going to be unipolar, leaving their cell bodies outside of the CNS. Interneurons are completely embedded within the CNS, so the axonal terminals of the sensory neurons are in the CNS, and the whole interneuron is in the CNS. And most motor neurons, we will see that their cell bodies are also in the CNS. Of course, if we add the second in a row autonomic motor neurons, then we would have some cell bodies of them in the PNS, as I illustrated earlier. Those second autonomic motor neurons we refer to as postganglionic. So finally, here you see a nice comparison of somatic versus autonomic motor neurons. And there's a little bit more information in here than you really are ready for in lab. In lecture, you will figure all of this out. But so here we have the central nervous system illustrated in the blue-gray. Here we have our effectors, right? And then what's in between is basically representing the peripheral nervous system. So somatic motor neurons always have their cell bodies with their dendrites in the CNS, long axon, making it all the way to the skeletal muscle. For the autonomic nervous system, we typically see two neurons in a row. So here we see the sympathetic and parasympathetic division. There is a bit of a different thing going on in the sympathetic nervous system because the adrenal gland gets involved, but really the adrenal gland, the inside, is really just a bunch of these post-ganglionic neurons. So in the autonomic nervous system, we call these preganglionic neurons, and these guys post-ganglionic neurons. Remember, autonomic motor neurons have very different effectors, right? So we have, again, a lot of cell bodies in the CNS, but indeed there are some cell bodies of the autonomic nervous system in the PNS, namely the cell bodies of the postganglionic neurons. And when there's many of these cell bodies here, we form a ganglion. From there, the term preganglionic and postganglionic. And so that wraps up our discussion for AP1 Lab on the introduction to the nervous system and the discussion of nervous tissue.